Okay, ladies and gentlemen, you are all welcome to my YouTube channel, and today we are going to look at the concept of lipid digestion and absorption. So what is a lipid? Lipids are generally large molecules, but they are not as polymer as carbohydrates and nucleic acids. Because, of course, if you look at carbohydrate and proteins, they are the polymers of simple sugars and amino acid. But in the case of lipids, it's just an ester of fatty acids and glycerol. And of course, they are large molecules, but insoluble in water, but soluble in organic solvent. So generally, lipids, the best way to define them is based on their solubility, that they are soluble in water. Sorry, they are insoluble in water, but soluble in organic solvents. So that is the best way to define the lipid. Because of the complex heterogeneity nature of them, you can't define them based on their chemical composition. And of course, most of the enzymes that are involved in the lipid digestions, they are the water-based enzymes. They are water-based enzymes. But now the question, how does the human body digest the lipids? So like, for example, in the mouth, we usually chew and emulsify the lipids. So the lipids cannot actually be digested directly the way they are because of their insolubility in water. So most of the digestive enzymes, they can't actually work on them. We can't actually digest them. So as a result of that, we need emulsifying agent that will emulsify them to make them more water soluble. So chewing and emulsifying phospholipids enable digestive enzymes in the mouth called lingual lipase to do their work. And of course, when these lingual lipase together with chewing and emulsification work, then a lipid droplets will be generated, which actually a product that separates the water component or the water soluble component from the water insoluble component. So at the end, we'll have something like the lipid droplet. So in this case of lipid droplet, you will see that in the surface of this lipid droplet, you will see that all these components like RAP18, urolipin, steroids, sterol, CCT, and phospholipids, they are all water-soluble components of the lipids. So therefore, when you say droplet, it means that all the non-polar component of the lipid will remain hidden, will remain inside, while the water-soluble component will be exposed. So generally, in the lipid digestion, the digestion in the mouth and in the stomach is just like a preparatory piece of the digestion. It's like it is just trying to prepare the lipid for the digestion. So then, so of course, the digestion of lipid usually takes place in three major parts. It takes place usually in three major parts. It takes place in the mouth, small in the stomach, and the small intestine. But generally, the digestion in the mouth is done by the limited enzymatic digestion. It's just a limited enzymatic digestion because it does not actually fully digest the lipids. So that is why they are called limited enzymatic digestion. So the limited enzymes in the mouth is the lingual lipase, while the limited enzymes in the stomach is the gastric lipase. And of course, the major digestion of lipids is taking place in the small intestine, where emulsification, the full digestion, usually takes place. So now let's actually look closely on how this lipids is being digested. So the major dietary lipid we take is, of course, we have triacylglycerol, which is the more than 90% of the lipid that we take. And we have cholesterol, phospholipid, and free fatty acids. So these are the three major dietary lipids. And then the average normal Indian diets take like 20 to 30 grams of lipid per day. But in the Western diet, it is usually 2 to 3 times that of the Indian diet. So, and then the next thing that we are going to look at after the major sources of our diets is okay, so the neutralization of lipids are usually insoluble in aqueous solution, but the digestive enzymes are present in aqueous medium. So this leads to certain problem in digestion and absorption of lipids. So as we said, actually the lipids is usually insoluble, and of course the enzymes are in aqueous medium. So to digest the lipid, it becomes problematic as well as the absorption. But how do you overcome this problem? It's through increasing the surface area of lipid for digestion. When we say to increase the surface area, it means to break the lipid into like smaller particles, and then of course emulsification of lipid by bile salt, and then solubilizing the digested product for absorption. So these are the three major ways of overcoming the problems of lipid digestion. First, we have increasing the surface area first of the lipid, and then second, the emulsification of the lipid, and then solubilizing the digested product. So these are the three major reasons or the three major ways of overcoming the lipid digestion. So now the digestion in the stomach. So the heat of the stomach usually liquefies the lipids, and emulsification of the lipids start in the stomach by peristaltic construction. Of course, there is a peristaltic construction. So the peristaltic construction is involved in the digestion of it's involved in the emulsification of the lipids. So it means that peristaltic movement is involved in the emulsification of the lipids. And then of course this lingual lipase is an enzyme that found on the tongue, that is from the mouth. And of course, since here we're talking about the stomach, so the major enzymes that found in the stomach for the lipid digestion is gastric lipase. And this gastric lipase, it acts on the short-chain triacylglycerol. Also, the lingual lipase is also the enzymes that also act on the short-chain triacylglycerol. Of course, the short-chain triacylglycerols are usually found in milk, butter, and ghee. These are the types of the food products where the short-chain triacylglycerols are usually found. So that is why these enzymes, lingual lipase and gastric lipase, it's usually more active. It's usually more active in infant. So the action of this lipase is significant in newborns because the major food product is milk, and that is why the enzyme is more active in the newborn babies. And then the next thing is actually the digestion in the small intestine. So first, the digestion of the small intestine is first started with the hormones. There are two major hormones that are involved, or that are very, very important in the digestion of lipid in the small intestine. And of course, the first enzyme is called cystokinin. It's called cystokinin. And this cystokinin is abbreviated usually as CCP. So this cystokinin is an enzyme that actually stimulates the secretion of pancreatic lipase. It stimulates the secretion of pancreatic lipase. It's an enzyme that it's a hormones that's found in the small intestine, and the major functions of the hormones is to stimulate the secretion of pancreatic lipase. And of course, another major functions of this cholecystokinin after secretion of pancreatic lipase is also involved in bile secretion. Bile secretion. And after bile secretion is also help in slowing the relief of gastric content. Slow the release of gastric content. Of course, remember that we said that the digestion of the lipid first started in the mouth and then in the stomach. And of course, in the stomach, the content there is highly acidic. So this cholecystokinin helps in the slow release of the gastric content into the small intestine. And of course, it's involved in bile secretion and then pancreatic lipase secretion. So this is the two major work of these enzymes. And then these are the three major functions of this hormone. And then another hormones that is also found in this small intestine is another enzyme called secretin. Another hormones called secretin. And the major function of this secretin is the release of watery soluble rich in bicarbonate by the pancreas. So it helps in the release of bicarbonates by the pancreas. So now, what actually happened after the release of these enzymes, bicarbonate, by these two major hormones? First, there is emulsification of fats, and then second, there is an enzyme's action on the lipids. And we are saying emulsification. What is emulsification? It's a process of dispersion of lipid into smaller droplets by reducing surface tension. And emulsification is essential for the digestion of lipids. And this emulsification is usually done by bile salt secreted by cholecystokinin and of course the peristaltic movement of the lipid. And then of course, what the bile does is the bile salts are called biological detergents. They are called biological detergent, and they help in the emulsification of lipid by forming micelles. So they help in emulsification of lipids. So once they emulsify the lipid, then they will produce what we call a micelle. And of course, this bile acid is synthesized from cholesterol. So we produce bile salt from cholesterol in the liver, then we store it in the gallbladder. When it's synthesized in the liver using cholesterol, then it's now stored in the gallbladder. Store it in the gallbladder. And then it's secreted while bile in the small intestine. So secreted with bile in the small intestine. Of course, so whenever there is a lipid, then the liver will now secrete the bile into the small intestine. And chemically, these are, when we said bile salt, they are called sodium glycocholates and then sodium tauricolates. Sodium tauricolates and sodium glycocholates are the chemical nature of the bile salt. And along with bile salt, phospholipid also helps in emulsification. So apart from the bile salt, the phospholipid also help in the emulsification of fats. So it means that in this case we have three things that help in the emulsification of fat. First, the bile salt is the major detergent, and the first one is the bile salt, and then the second one is phospholipid, and then the last one is peristalsis, peristalsis movement. So these are the three ways in which the emulsification of fat takes place in the body. And then, so now what happens in the digestion of fats in the body? And of course, the digestion of the lipids is done by the pancreatic enzymes. And these pancreatic enzymes, we have three major of these pancreatic enzymes. Number one, we have pancreatic lipase, we have cholesterol esterase, and we have phospholipids. So these are the three major pancreatic enzymes that involve in the digestion of fat in the small intestine. So now let's look at how the triacylglycerol is hydrolyzed by lipases. So first we have triacylglycerol, and then this pancreatic lipase will now remove the fatty acid at carbon 3. So once it's removed, then you are now left with, of course, it is called triacylglycerol because there are three fatty acids, one here, another one here, then another one here. So we have one here, we have the second one here, and then we have the third one here. So now what happened is this pancreatic lipase first act and remove fatty acid at carbon 3. That's left with diacylglycerol, and then still another pancreatic lipase act on the diacylglycerol and then removed the glycerol at carbon 1, leaving it with 2 monoacylglycerol. Then through isomerization, then it will now become 1 monoacylglycerol. So it means that this phospholipase cannot be able to remove fatty acid from carbon 2 till after it now isomerize it and change it from position 2 to position 2. And then the phospholipase, then the pancreatic lipase will now remove the fatty acid at carbon 1. Then lastly, at the end, after the digestion of this pancreatic lipase, then we'll now end up of 1 free glycerol with 3 free fatty acid, with 3 free fatty acids. So after this digestion by the pancreatic lipase, then the next one is of course the digestion in the small intestine. So the digestion of dietary phospholipid is usually done by phospholipase A2. So phospholipase A2 hydrolyzes fatty acid at the 2 position of glycerophospholipid, produce lysophospholipid. And this lysophospholipid being detergent help in the emulsification and the digestion of lipids. So it means that lysophospholipid is also the phospholipid that majorly help in the digestion of lipid in the small intestine. And then of course this is the enzyme phospholipase A2. It removes the free fatty acids at carbon 2, and then we generate lysophospholipid, which is the detergent that are used in the digestion in the emulsification of fats. And then the next one is the digestion, of course, still in the small intestine, using the pancreatic cholesterol ester hydrolase. So it's the enzymes is actually involved in the digestion of cholesterol ester. So cholesterol esters is also another class of lipid. So this enzyme cholesterol esterase will now hydrolyze the cholesterol esters to produce 1 free fatty acid and cholesterol. And that is the 3 major enzymes that involve in the digestion of lipid in the small intestine. So that is where we say the digestion of lipid is majorly take place in the small intestine. And then what are the products of the digestion? So following the end products of the lipid digestion, we have free fatty acids. We can also generate 2 monoacylglycerol. We can also have 1 monoacylglycerol. We can also have free glycerol, free cholesterol, and then lysophospholipid. So this together with bile salt form a mixed micelle. Fat-soluble vitamins, that is vitamin A, D, E, and K, are also packaged in this micelle and are absorbed. So in the next video, we are going to look at how lipids are absorbed. So this is the digestion of lipids. So the major aim of the digestion, what we are trying to achieve at the end of the digestion is to get the end product of the digestion, which are listed here. And this end product of the digestion will now all combine together with the bile salts to form a mixed micelles, which will be actually transported to the blood stream. So in the next video, we are going to look at how the lipid is being transported to the blood stream. Thank you very much. And if this is the first time you are coming to my YouTube channel, please subscribe. Thank you.